Good evening, everyone. It's really a pleasure to be here, and a little bit of an intimidating situation because, as you know, Ilan Samish, who was supposed to be here and introduce David, is not here. And I don't know if you ever had the experience of getting a presentation to present that you did not produce yourself. It's a first for me, and it's intimidating. So I apologize in advance. These are not my slides, but I also thank Ilan for preparing a presentation to introduce David. So it's really a pleasure to have tonight David Shaw, who is the Warren Delano main 3D SIG speaker and the Warren Delano session speaker. And so who are you, David Shaw? Again, these are not my slides. Now, David has a BSc summa cum laude from UCSD and a PhD from Stanford, and then he has a faculty from Columbia, and that has involved many appointments as a faculty of computer science, professor in biochemistry and molecular biophysics, and a senior research fellow at the Center of Computational Biology and Bioinformatics. He's one of us. But he has a few more things on his CV than most of us. So he has a few non-academic research appointments, non-academic and research appointments. So from 86 at Morgan Stanley as VP of Technology and Tartaglia's automated proprietary trading, as well as then funding D. Shaw, who all of you here heard about, and which is a company that has been described as the most intriguing and mysterious force on Wall Street. That sounds intriguing, the discussion itself. Now, what do you do when you're 50 and you got everything? You do science. And D. Shaw really is pushing the envelope on molecular dynamics. So really, I apologize if I'll oversimplify it. The whole idea is to build specific hardware that is specific for the task in this case, and with that greatly speed up things. And by having architecture that is specific for molecular dynamic simulations to calculate the most time-consuming step, which is the energy evaluation, then you can speed up things a lot. Now, I guess you don't have to go see this YouTube video. Probably David will speak about it today. But really, just like Antony van Leeuwenhoek created the microscope, and that technology opened the door to many things, Anton, and I'm not sure myself by my ignorance if Anton comes from the same. Yeah, it does. Okay. Ilan knows more about this than I do. But really, the computer, Anton, is the technology that drives discovery, and it opens the door to inquire several biological processes that were not possible before, just by the fact that we were not, I say we as a community, we were not able to sample these processes long enough to see things happening. And it's been proven over and over again in all these wonderful papers that we all read. And well, David has been recognized by this in many ways. He's been part of the Council of Advisors on Science and Technology for the President of the United States. appointed by Clinton and Obama, and has been elected to the American Association of Advancement of Science, American Academy of Arts and Science, National Academy of Engineering, and National Academy of Sciences. That's an impressive CV, and we are really fortunate to have been able to lure David to 3D SIG after many, many years of inviting him. Finally, we managed, and it's really a pleasure to have you here, and looking forward to your presentation. Thank you very much. So sorry for any mistakes.
Oh, that was great. Perfect.
Do you want to use this thing or
Oh,
just this
that would be great if I could. Put the,
pockets?
yeah,
I think it works. Tested it before. But this will pick up anyway.
yeah, this is better. Okay, does this work? Great. Well, let me first just start by saying it's an honor to be here, especially because it's an honor to be at a session that was named and created for Warren Delano, who was an amazing guy. I remember the day my friend and colleague Rich Friesner told me that he had died at an incredibly young age, and everybody was really stunned. So being able to honor his memory is a wonderful thing. Oh, I think I'm just connected through here. Oh, it's not working, or? No? Okay. How's that? Good, I'll do that. Just yell at me if I forget to look at it. I do that sometimes. So let me just start by talking a little bit about what we focus on. And I'm going to just take a few minutes because there's some people in this room who don't, well, many who don't use molecular dynamic simulations, which is what we do, and then going into the guts of it to try and understand what part is hard, what part takes a long time, is just useful by way of background, because I'm going to be talking for a while about why things that are very long, why simulating for a long period of time can give you different snapshots on what's going on. So everything that we do is really focused on dynamic simulations, using MD simulations. And what we're interested in the long run is to study the structure and the dynamics, in particular of proteins, with a focus on understanding, at a scientific level, understanding the mechanism by which various physiological processes, both normal ones and pathological ones, are mediated by various kinds of structural changes. And then the very long-term goal of all of this, and the reason that I was interested in switching over, and the reason that most people in our group are there, is to try and help cure people at some point. And at some, I guess later on, toward the end of the talk, I'm going to be talking just a tiny bit about what some of the possibilities might be for doing that, for aiding that process. But also some of the potential limitations, and at this stage, the jury's out. We just don't know the extent to which MD simulations, long MD simulations, are going to make an impact, and if they do, to what extent on the process of drug design. But that's the hope, and so a lot of what we're doing is focused on that long-term goal. Our group has a very long-term focus, though. We're not so much interested in what we can do this year, because we would, but there's not that much we think we can do this year, but we really believe in pursuing the basic science, trying to understand both physiologically what's going on, and also in terms of methodology, where could something like this contribute to that process? So our tool is all-atom MD simulations, and I'm just going to start by giving you an example of one of them. I'm going to start it because this one deliberately lasts a while, especially if it gets started at all. Oh, wrong one. Okay. There we go. So this representation will be clear to everybody in this room. We've got a bunch of balls, which are atoms. And I should mention that this simulation is sort of smooth, because otherwise with a simulation of this length, you'd see all vibrations. It would be a fuzzy mess. But the kinds of things that we're interested in following are these big structural changes. There are a lot of interesting things going on where you have little rotations of torsion angles and vibrations and so forth. And well beyond the point where we could simulate, we as a field could simulate for this long, there were good things that were learned there. But when it comes to looking at these big structural changes, this is not a particularly interesting protein from our viewpoint. It's GPW, and it was heated up to the point where it would be at right around the melting temperature, so it folded and unfolded. But those big structural changes are the kind of things that we're interested in. So now I'm going to, oh my. Okay, I may plead for help with Lucas. Do you know what's up here? Yeah, I'm not seeing the other slide anyway. I'm just seeing one.
It looks like it's
Not
not in here.
in there.
You can
Okay,
do it
well,
manually, just
yeah, I'll have to do it manually. I hope it's not the wrong version of this, but in any case, let me just by hand show you this was on Anton. It was an Anton simulation. The movie you saw was 44 seconds long, I think it was, and if we were doing this on the world's fastest supercomputer, which was the state of the art, the longest simulation that was done before the first Anton, it would look something like this. But you didn't even see it. It would be a quarter of a second of elapsed time here. So let's see if I can do that again. Nope. But what you would have seen if you'd seen that is there would be something going on. It would be smoothed out, but you wouldn't see any of those structural changes at all that we're interested in. So we were interested in doing things much, much faster, or aiming for a couple orders of magnitude faster than that. And the thing that's hard about that is that, actually those slides showed a little bit of it, but the process of MD simulation involves lots of time steps. It's integrating Newton's laws of motion, but doing that numerically involves over and over again at a femtosecond resolution of time scale, which is less than the time that you have for a bond vibration, repeating that process. And for us, we were interested in the millisecond time scale. So that's doing it on the order of 10 to the 12th times. So anything you do that much is going to take a huge amount of computer time. And the things you have to do are conceptually take a look at any given atom in your simulation, look at the interaction between that atom and all other atoms in the system. Now, you can cheat a little bit for things that are far away and use various sorts of continuum approximations and solve it using finite difference, or simulate it using a Fourier transform and multiple methods, a variety of other things. But conceptually, if you look at what takes all the time, it's these quadratic number of pairwise particle interactions. And then for each of them, you execute the value of a force field. That was also shown there. And in particular, the part that takes a lot of the time is the non-bonded interactions, the interactions between atoms that aren't covalently bound to each other, or even within 1, 2, 3 links of each other. But you're looking at Coulomb interactions, just oppositely charged particles attract, and if they're same charge, they repel. And then this one is, whoops, a little bit funny. I think a lot of you know what this is about, the van der Waals interaction. And basically it has two terms. This 1/r to the 6th one basically says that independent of charges and all that, atoms like being close together, but the 1/r to the 12th says not too close together for fairly odd and weird quantum mechanical reasons. The 1/r to 6 takes some time, can't be ignored, but this is the part that takes most of the time, just the attraction and repulsion of distant atoms. So once you've done that, it's very straightforward. Each one of these atoms, you just move it in the direction of that force by a tiny bit, repeat it 10 to the 12th times, and you're done. So before Anton, the longest simulation done was about 10 microseconds of biological time. And the problem is, from our viewpoint, that we were really interested in the kinds of conformational changes that were biologically and potentially pharmaceutically important. And a lot of that, took place on timescales between 10 microseconds and 10 milliseconds or so. And that included protein-protein interactions, interactions between proteins and nucleic acids, although we haven't done too much of that yet for reasons related to force fields. I'll talk about that. Interactions between proteins and various kinds of ligands, including endogenous ligands, ones that are in your body already, small organic molecules that are there, or drug molecules. And one of the things that's quite interesting is, which we're spending some time on now, is looking at conformational changes in a target protein, one that it causes a disease, for example, that are induced not by binding to the binding site that's relevant to the activity in question, but to some other site on the same protein, the so-called allosteric changes. Attach something here, something else changes shape, and then it affects a signaling pathway or some other function. So these are the kind of things that often take place in that timescale that was well beyond the range of previous technology. So we designed a special purpose machine for that called Anton. This is Anton 1. You can recognize it by the fact that Anton is in red, as opposed to Anton 2, which is in green. Otherwise looks the same. Anton is a special purpose machine. The main part of it that has to be made fast is particle-particle interactions, although it turns out if you speed that up dramatically, then other things start to become significant enough that you could ignore them before, and now you have to make sure those are fast enough too. But that's the core bread and butter. And the machine was designed together with an algorithmic approach that was somewhat different. You couldn't just speed up the hardware and keep the algorithms the same. The two had to be done together. It's been operating now since October 2008. When Anton 1 was put in place, it was about 100 times faster than the fastest supercomputers available there at that point. And that allowed us to do the first millisecond scale continuous single simulations at an atomic level of detail, including the water environment, which is represented explicitly as individual water molecules, which is something that was important to us because a lot of biology actually is influenced by the relationship between the first shell of water, as one's close to the protein, and the surface of that protein. And then we designed Anton 2, which is reused the cabinet at a different color. That was deployed in, seems like more recent, but it was actually 2013. And that is, depending on the size of system and so forth, another 4.5 to 10 times faster than Anton 1. So now we do multi-millisecond simulations pretty much routinely. And in fact, one simulation, may have two at this point, I'm not sure, but one is more than 10 milliseconds long. I should mention, many simulations that we run are much shorter. So there are a lot of things you can test in 10 microseconds or less. You do those very quickly. And that's still, actually quite useful to be able to do a single simulation really fast, just in terms of workflow. If you're sitting there and you can quickly run a simulation, say, oh, that's what's going on, or this setup doesn't work, this all falls apart, you can move faster. But the key thing is being able to simulate for long periods of time with a single trajectory. Now, that's distinct from another problem, also important, which is pure sampling. And there are a couple different categories there. One is running a large number of MD simulations with ordinary MD, but running them for each one for shorter periods of time. Now, that's very useful. There are an awful lot of things that you can do looking at a lot of simulations and collecting statistical results, and also with various kinds of more sophisticated techniques like building Markov models, where you look at transition probabilities and gain information about that from large numbers of short runs. It's a very, if I were a funding agent, I would support that research very enthusiastically. It's just different from what we do because there's another class of problems where you really have to look at a single long trajectory in order to gain certain sorts of information. And let me also just mention another one that, again, is a different sort of problem, which is not only can you have a lot of short simulations, but another possibility is using various kinds of enhanced simulation techniques, enhanced sampling techniques. And in most of those cases, you're not actually seeing a realistic trajectory, not just no single trajectory, but the way the molecule moves won't be an actual trajectory, but you can collect some information, rigorous statistical information, because these various algorithms sample the Boltzmann distribution, the occupation of various conformational states, various structural states, follows the distribution, with an exponential in there. It tells you how long it's going to be in residence within each of those states. Very powerful. It's an area under active development. It's one I personally believe in, as do other members of our group. It's just not the problem that we work on in our group. So this is, maybe this is the one. Yeah, this is the one with this. Okay. You've already seen this. It's not showing you anything, but I'm just going to run it because it's prepared. Okay, we'll skip that. So this is what it looks like. That's as long as you would see in the same simulation running on a regular supercomputer. So you see different sorts of things. Quick ad, not for us, but for recruiting other people to the field, which is we donated an Anton 1 machine to be available to the research community, the not-for-profit, non-commercial research community in the US for various sorts of structural and regulatory reasons. And that's been available since 2010, I guess. And so far, 133 independent research groups have used that to run Anton 1, and now it's starting to be Anton 2 simulations. We don't allocate time on the machines, the National Academy does. But it's great to have people doing it. They think of things that people in our group wouldn't have occurred to them, and they do outstanding work using specialized expertise, knowledge of particular proteins. And I would encourage any of you who might be interested in doing that to apply for time on that, and every year time is allocated. So let me just give you a few applications. I'm going to start with some various things that we've done that actually work, things where we've been able to discover some things, sometimes subject to experimental validation, but as usual in science, sometimes you would view it as a hypothesis that has not been refuted by some evidence, but you never know until you start working on it whether that should be refined or even rejected at some point. But let me start with that and then tell you some things that don't work and show you examples, which I haven't had a chance to talk about as much, but because we gained some of this information over time. But interestingly, there are things that don't work in MD simulation because of the qualities of the physical model, apparently, but that you have a difficult time in some cases detecting. You don't see the problems unless you can run for a very long period of time. But first the good news. So we've studied protein folding for a period of time, partly because it's of scientific interest, but also it turns out that there are a number of diseases that, even in the last 5 to 10 years, we've learned that there are more than we had thought before that are attributable to the misfolding of proteins. And various bad things can happen in this case. The sort of breakdown I'm putting here was really something Susan Lindquist laid them out in this way in a paper, and I think it's a very nice way to think of it. So one, probably the best known, is that sometimes when a protein is misfolded, it can aggregate. You can have large numbers of that same protein bind to each other and form fibrils, and things get tangled up and you have a mess, most famously in the brain. There are various neurodegenerative diseases that are now known to be attributable, at least in part, to misfolding. You can also have something where if a protein is misfolded, it gets degraded in a way that it's not supposed to in the body. Often something where it's supposed to get degraded, but it gets degraded so quickly that it can't serve its biological function. Another thing that can happen is you can have a misfolded protein that would work, except it's sent to the wrong compartment in the cell because of some part of the structure not being able to get attached to a carrier protein or something else. And then one of the other ones, this is probably best known in the case of Alzheimer's disease, but it looks like it's present in a number of other diseases, is that some of these structures look like they can be toxic. We know certainly in vitro that a number of them are toxic, and in some cases there are intermediate states that may be the source of some of the toxicity or fragments of those proteins that are cleaved off. But in any case, misfolding can translate into poisoning various cells and killing them off. So there are a lot of reasons to study folding now. And one of the interesting areas now is to study not just folded proteins. Most proteins in the body assume a characteristic native state in the body, but not all. There's some that either are unfolded until they find a binding partner, or interestingly, some cases where you have a folded protein, but there's part of the protein that's unfolded and that can not only assume a particular shape when it has a binding partner, but assume different shapes depending on what that binding partner is. So studying unfolded states is extremely interesting, and as I'll mention in a little bit, it's also something that MD has gotten really, really wrong, and so it's an area of active research. It could also be that, we had done some work, I can't remember exactly when, but where we took a characteristic set of proteins and were able to fold it in the simulation for the first time, all but one, and that turned out to be not very thermostable, but just starting it from an extended conformation, it folded into the native structure. You can imagine using that for structure prediction, but it's probably not a very good idea at all. There are probably better ways to do that, even computationally, than running a whole long MD simulation. We did it really to learn some things about the folding process, which turned out to be quite interesting, and I guess I'll get to that now. So let me show you this one. I think it was actually the first one we did, but it's also the protein that 10 microsecond simulation was done on to try and see the folding of a particular WW domain called FIP35. And this is a relatively short extract from the middle of a very long simulation. And what you see is in gray, that's the native state. We started the simulated protein in extended state, and it forms, I won't talk much about how it does it because I want to make sure not to run out of time, but in a characteristic order, there are two beta hairpins. They actually don't, it doesn't reach the one that you see. It reaches the wrong one first, but then that's not thermodynamically favored, so it flips around, undoes that, and always seems to come back to one of those hairpins. Then the other one folds in a certain structure, and we were interested in, one of the questions was, when folding occurs, does it have a characteristic pathway? It's always microscopically different because this kind of simulation is a chaotic process, but does the overall folding pathway, do the key events take place in the same order? And for the most part, we found that they did, and learned some other things about that. Now I'll just summarize this quickly. We used some of the things we learned in studying it as a sort of validating step to see if we could design a protein, a variant of that WW protein that would fold faster. And we made, I believe it was 3 amino acid substitutions. And then Martin Grubela tested the one that we had, and it did fold about 4 times faster, which is now the fastest folding, I guess, beta structured protein that's been done. It's just a way of looking at whether you understand what's going on to try various things, and if it didn't, we would worry about that model. Here are the 12 proteins that we folded, and I have said enough about this. Now, those were all, this is important to emphasize, the ones that I showed you, those 12, they were all small, fast-folding proteins, and actually some of them were big enough that that wasn't the key thing. The main one is that they were selected and in many cases designed to fold quickly, just because that allowed you to study them this way. We then simulated a couple years later, so one that had a more complex folding landscape, and that's ubiquitin, which is also very well studied, which was helpful to have a bunch of experimental data. And then we did a long simulation, over a millisecond simulation, And that one was done, these other ones I mentioned were done at biological temperature, so you would see them driven downhill to the folded state. In this case, we simulated around the melting temperature, so we would see a large number of folding, by definition, of folding and unfolding events, which we could study. And I'm just going to show you one of those transitions to get a sense of what you can learn. So ubiquitin has, let's see, 4 big beta hairpins and then one small one. It's got an alpha helix, a little 310 helix. But the question was, what forms first and why? What's the hard part here? What constitutes that transition path? So this one slowed quite a bit, just so you can, smoothed quite a bit, so it'll look not like a usual MD simulation, but you can see what's happening. So very early in the game, you're going to see some of this beta sheet forming. The hard part is at the other end, right over here, there we go. It occurs in the middle of the beta sheet, not at the end. And so getting that over there is now here is the hard part. And it's actually misregistered at this point. It's in the right place finally, but it has to, find its way up. So the big thing that slows it down, what's happened is as soon as it's there, all of this happened fast. You're going to see this helix form. There we go, strand. This is the 310 helix, and a tiny little beta strand at the end falls into place. But I won't bother running it again for you. But the key thing is that there's one event that has to happen, and as soon as it does, everything very quickly falls into place. And the part that did happen in this particular case, it's different for different proteins, but in this case, it was overcoming the entropic barrier that's involved in having this thing floating around at random. It has to find its way just physically into the right location. Once it's done that, it constrains the conformation to the point where it finds the native state very quickly. So this is the kind of thing that we do with a long simulation, which is very hard to do with sampling. You really want to see order of events and mechanism of folding. What are the driving forces in, entropy and enthalpy that drive the process, and so forth. And I just had added this recently, something to talk about some of the implications of protein folding and protein folding intermediates in terms of pathology. In one case, Alzheimer's disease, we've known that it was associated with amyloid beta peptide and various things going wrong there. It's, at the very least, for a long time, we've known on autopsy that there were tangles of this material, amyloid plaque, that's found in the disease of people with Alzheimer's disease. One of the things people have been looking at, more recently, including David Eisenberg, with whom we collaborated on some of this, was toxic oligomers that were fragments of amyloid beta that seem to form structures that we believe may be toxic in themselves. And it's hard to look at that experimentally. He was able to learn various things about it, but in the case of amyloid beta, first of all, there are a bunch of these oligomers, and they don't last very long. Very quickly, these fragments wind up being part of the full fibrils, and then that forms all of the stuff that gloms together. So there was no previous atomistic structure known for an amyloid beta fragment. There was, however, a different one, which is called alpha-B crystalline, which is a very similar protein, and David and his group were able to see that one happens to move more slowly, so it was long enough that he could get a snapshot during that period of time and see what the structure is. And we suspected that amyloid beta would go to a similar sort of structure, and that's what happened. So we looked at a C-terminal fragment, the last 10 residues. It's, I think, 11 in the case of alpha-B crystalline. So here is, let's see, okay, in gray is the known crystallographic structure of alpha-B crystalline, and then here are individual amyloid beta fragments which are going to form into a barrel structure. they grab together, and let me show you what happens. So they wander around for a while, and there's sort of a nucleating event, and then very quickly they go into a structure that really is very close to the other one. So it's not a surprising result biologically, but because that looks like it's, there's evidence that that may be a pathological structure, it's an interesting thing to study. I'm going to show you another thing. No, actually, I've eliminated one that had to do with Alzheimer's. I'm giving a different example. So let me just move on to a different application where interesting things were discovered. This is one of the positive cases. And that's we've done some work on various ion channels, but I'll be showing you a potassium-gated ion channel. Sorry, a voltage-gated potassium channel. And in particular, we studied KV1.2. The way that works is basically it's a tube that goes through the membrane. It either passes potassium ions selectively if it's in the open configuration, or else it closes up and no ions can go through there. And the voltage gated means that that's determined, whether it's open or closed, is determined by the voltage across the membrane. So the channel is closed when you have a hyperpolarizing voltage, so it's much more negative inside the cell than outside. That's called the resting state. And then at depolarizing voltage, it's the opposite. The channel opens and the potassium ions flow from the inside to the outside of the cell. The voltage is the opposite. And there are various mutations. People who are born with different kinds of inherited channelopathies can have problems with conduction, electrical conduction in the heart, certain chronic pain conditions, certain types of paralysis, some types of migraine headaches. So we were interested in how it was controlled, but also, potentially, is there something that can be done about it? So let me just start by showing you the permeation of potassium ions through the channel. And this one just shows the pore domain. There's a voltage regulatory domain that senses the voltage. It transduces a signal in a way that we looked at. And then what it does is open and closes this tube. So this is just the tube itself. The big colored balls are potassium ions, which are colored differently just so you can watch them move because they snap pretty quickly. The fact that they get stuck in known sites here was known before, various carbonyl groups that sort of trap them. And we just looked at whether it went through there and what the rate was and some other things. But the more interesting part to us, once we were done with that, was to answer the question, how does the whole thing work when it's regulated? And what's the sequence of events going around the cycle between opening and closing? So this is the structure of KV1.2 that Rod McKinnon discovered some years ago. And it's a tetramer, and they're colored, each of the monomers is colored in a different color. This is the part we saw. This is the pore domain, and these things falling off the sides are voltage sensors, but the voltage sensor domains. What was known before was the open state. That's what we had just seen. So there was a snapshot of that state, but the closed state had not been observed. And there were various models of what the structure of the closed state might look like. And also, more interestingly, the process of opening and closed wasn't understood. There were some pieces that were understood, but the whole process was something fascinating and not understood. And in particular, how does that voltage difference how does that induce those structural changes that cause it to be open or closed? So what we did is to run a long simulation, which I won't show you, but just in terms of the results, we were able to simulate by simply having a simulated charge with different ions on the two sides, simulate different voltages across the membrane, run this a number of times and look at what happened, try and analyze the individual structural changes. It's not just a matter of looking, you have to measure all sorts of things, but pretty quickly we were able to get a sense of what was going on. So this is tricky because the cycle is, let's see, can you see this? I'm going to try one other. Hey, that's better. So it's going in a cycle, but the inside of the cell in each case is at the bottom, and the outside is over here. So this is the known open state, and these little green things are the potassium ions. So now what we have is the inside of the cell becomes more negative. So what happens? Well, it was known that there are 5 charges, positive charges up here on this helix. And so naturally, if this becomes more negative, there's a downward pull, electrostatic pull. So that pulls this guy down. Now there's a linker domain here. And when this goes down, it sort of releases the tension on that linker. It turns out this thing right here, it's 1/4 of the pore domain, but it just, and illustrated in a way that's not quite right, but I did my best with Adobe Illustrator to sort of, use a little artistic license. But this is sort of spring-loaded. Now it's free by this relaxation to close. And in addition to that, there's another effect, which is that we had some potassium ions sitting in this little vestibule here. When the charge down here becomes negative, it pulls these potassium ions, which are positively charged, down into the cytoplasm, and that leaves this waters here. Now, it turns out these two sides are highly hydrophobic, and those waters were only in there sort of to escort the potassium ions and neutralize some charge. And so when they're stuck in this greasy surface, they want to get out. That leaves a vacuum. Nature abhorring a vacuum, that plus the freedom here causes this to close up. And along the way, this guy is going to pull the helix here with the charges, pulls more into the center. And pretty soon what we're left with is something that we believed was the closed state. And it's consistent with a bunch of experimental evidence. There's some things that are certainly arguable and have been argued, and we're not sure of those details. Nobody is at this point, but it provides an explanation, and some parts of it look like they're likely to be right. And just a quick one. It's known that certain pathologies or certain sorts of mutations cause different bad things to happen. What you're seeing here is, it looks like it's the pore domain, but it's actually part of the voltage sensor domain, which has no business opening up. Nothing is supposed to pass through here. With a certain mutation, you can't really see them, but until you run it. But with that mutation, turns out there's a leakage current. You're going to see where they are here. Big yellow ones are potassium ions. They are now leaking through the channel, and you can sort of see what's going on, which explains the fact that you're not maintaining the correct voltage and that it could cause problems. So now, let's see how I'm doing for time. I think I'm still okay, basically. So now I wanted to talk a little bit about some stuff that at least seems like it's more directly connected or potentially more directly connected to the problem of designing drugs to treat various diseases. And I just want to emphasize that this is highly speculative. Some of the things we've seen are pretty solid, and they're various, they match a lot of experimental data. But the question of how can this actually be used, to me, it feels like this has got to be useful in a lot of ways. And people who design drugs have said, oh, that's what's going on. That's very useful. But whether it can be part of a drug discovery pipeline and accelerate the development of drugs, that's a question that we just don't know the answer to yet. But it's sort of fun to look at what we can see anyway. So this is an experiment where it's SARC kinase, a well-studied kinase. That's a kinase, that's this protein. This is a molecule called PP1, which I believe was originally developed as a drug candidate, but didn't pan out as a drug. I think it may have been because of toxicity, although I'm not sure. And this example of the molecule is going to stay where it is, just as a reference point, but we're going to let the protein itself move around. And then the simulation was to release another copy of PP1, which is going to be the red thing, out here, and just see if it could find the binding site and how it explored the surface to find that known binding site. So let me see if I can do the simulation. So pretty quickly, PP1 is going to figure out that it's good to be close to the protein, which is not obvious, but Now it's going to, after a little time, wind up in, this is the ATP binding site, the catalytic site, and now instead of diffusing all around here, it's going to diffuse back and forth in the binding site and ultimately figures out that this is a good place to be. Even with current force fields, and this isn't always true, the state it comes up with is very, very close to the crystallographically known state. In fact, if you see it with the waters in here, it really fits very snugly where it should. And I'm just going to show you one or two more of these. Illustrates the same thing, but I enjoy seeing them, and also these are ones that actually do have to do with drugs. So this is SARC again, and the drug, excuse me, the drug, the ligand that we're going to see is an actual approved drug, approved for breast cancer, called dasatinib. And it again is going to find the right site over time. And it kind of gets in an area that's sort of the right area, but wiggles around until it finds a good location. This is more fun to watch in 3D with stereoscopic vision because it actually tucks itself in here very nicely. But it's another one of these examples where you don't need prior knowledge. You release the thing in simulation, you see where it binds. And it would be nice if you had candidate drug molecules and you saw an unknown thing and you saw it does bind and in a particular place. And that place may not be the active site, as in this case. It might be a different part of the protein that induces structural changes. So it seems like a very powerful technique, but again, how it fits into this overall process, from ideas to clinical trials, I don't know at this point. Just one more, no excuse for it, just to show you another one. This is the epidermal growth factor receptor, and it's another drug called lepatinib that's approved for breast cancer, and it finds its way into the right place once again. And this one is not a movie, but I'll just show you another example that's kind of interesting because of the way it binds. Now let me see, I have to refresh my memory about which one. I guess the blue is the crystallographic structure, and this is a case of its recognition of a protein-protein binding site, specifically interleukin-2 binding to the IL-2 receptor. And in simulation, what we found is that a particular ligand, and this is one that Sunesis came up with as a compound along the way. It was SP4206, where nice stuff has been published. And we found in simulation that it found the correct binding site. And in this case, the binding site wasn't fully formed at the beginning of the simulation. In the process of binding, it assumed the final state. So that was an interesting observation because we're interested in things that might have some of the characteristic of either induced fit, where a ligand actually causes the protein to fold around it in a certain way, or another protein causes it to, or a conformational selection, which is where, well, it would have gotten there anyway, but it's stabilized by another binding partner, for example. This is sort of a quick and dirty test. One of the things that we're doing now that we're trying to get a sense of what candidate molecules might be potential drugs. And we have started working on that over the past couple of years or so. And one of the things that we do is, a key figure of merit, one of the first things you look for is binding affinity, which basically tells you how tightly a given, ligand binds to a given protein target. So that's actually, how that thermodynamic quantity, how much of the time it's in the bound state versus not in the bound state is a function of free energy. It's a thermodynamic quantity. So you can't really study it just by looking at a single event in simulation and saying, well, this one falls off quickly, it must not be a good binder. You have to know about the on rate and the off rate. And that's particularly hard because something I didn't mention there, in those simulations I showed you of finding a binding site, the effective concentration was extremely high compared to physiological conditions. And the reason it's so high is that we're simulating a fairly small box of water, or water with solvent, with ions floating around. A small box with one molecule, and if you repeated that a lot of times, that would be the equivalent to a very high concentration. So under those conditions, the binding process happens pretty quickly, but if you looked at it in equilibrium, the amount of time you would expect to watch to see an unbinding event would be extremely long, impractically long. Now, there are other ways that you could get a measure of free energy of binding, and those are known techniques, and we've experimented with some of them. Different techniques like thermodynamic integration and so forth. That's a hard problem. This is just a quick and dirty thing where if you attach this thing and it doesn't stay there at all, it just flies off, it's probably not a very good candidate for a drug. So here's a simulation with a particular ligand where it's going to unbind very quickly. And in fact, one of the things it did that was pretty quick is to destabilize the structure. It moved this and allowed it to fly out. Here's another one that's very closely related, part of a congeneric series, where it's really stable, at least for the period of time that we studied it. Doesn't mean it's an nanomolar binder, but as far as a quick scan, I'm often asked, well, could you use this to measure affinity? And the answer is not really, but you can get sort of a sense of it. Okay, now here is a thing that you can do with MD. Hopefully it'll be useful, but it's something that's hard to do experimentally, and it's not practical to do with a short simulation. So this is a case where we took a look at this time, in this case as a phosphatase. Phosphatases, they're like the opposite of kinases, except they're really, really hard to target. They've been considered historically undruggable, even though there are a few successes there now. And the question, one of the problems is, well, in any case, it's very hard to find a binding site in the first place that looks like a pocket that a ligand could fit into and have a pharmacological effect. So in this simulation, what we looked at is just an apo simulation of the protein, SHP2, over a period of time, and then looked for whether, for some brief period of time, you saw something that looks like a binding site. And in fact, a pocket is going to open up right in here. I guess it's going to rotate and it'll be easier to see. So you'll see a deep pocket going right in there. Beautiful binding site, but it's not there most of the time. So the idea is to look for ephemeral binding sites, ones that are there, transiently for a short period of time. And then you can do other things. I won't describe what all of them are in the interest of time, but the idea then is to use this to find fragments that could bind there and ultimately design a drug molecule that could bind in those places. So it's a very nice technique. You would hope then that what you could do is use this to generate conformations that you could use, for example, using non-MD techniques. Conventional docking, for example, which is a much more efficient technique. It's possible to do it in a high throughput manner that, to a first approximation, what you do is take a given fixed conformation of a protein and then take all sorts of molecules, which might be like 1 million of them, and then move them by there and look at all of the different orientations, subject to some discretization, and see whether any of them seem to fit in there with a sort of fuzzy, very tolerant function, not with the full, I showed you the force field model that's used in MD. This is one that's a scoring function that's much more forgiving, and then find some that look like they might, they're not on top of each other, there are no obvious clashes. Ideally, you have a hydrogen bond donor matching up with a receptor and no charge conflict. And if you have an idea what the fixed protein is, the conformation of the protein, that could help apply docking. We played with that. We don't develop docking software, but we've used it to try and see whether that could, we could find target conformations and then do docking and then ultimately run actual experiments. But we don't know how well that'll work in the long term. Now, I just want to go through this is, again, highly speculative. What areas might be areas where long MD simulations could potentially contribute to the process of drug discovery? Some that seem like they probably should be able to work, some that are pretty far-fetched down toward the end. But then I'm going to move on to some things that don't work to close the talk. So where might you use it? Well, the most important thing still, I think, is the original area that we spent the most time on, which is understanding biological mechanisms at an atomic level of detail. Understanding, starting usually with a known structure, looking at that and then making changes, either mutations or simply watching over time or binding a drug molecule to something, to the protein, and then watching what changes happen to try and draw a picture of what's going on, like in the case of the ion channel, for example. Another one there, I guess I've leaked into that already, is to look at different mutations and see how that changes those things. And that would include both somatic mutations, things that are, you're exposed to radiation, some mutation changes and you wind up with cancer, or various kinds of germline mutations where you inherit at least a proclivity toward getting a given disease. Then another key part of it, and I think we've had some success there, is to try and understand how some drugs work at a mechanistic structural level, and also why they don't. So in a case of a mutation, there's some real success stories now in targeted therapies. Probably the poster child is, the first of those was imatinib. It's called Gleevec, which proved to be active and in a pretty stable way, a pretty durable response to a particular type of leukemia, chronic myelogenous leukemia, with a particular characteristic mutation, and it attached in a particular place and was able to block it. So, basically, the disease state is that you've got a kinase with a kinase domain, the catalytic domain, and then some regulatory domains. The regulatory domain can be locked up, and you get a fusion protein that's called BCR-ABL, where now you've got the kinase domain sending a signal that says replicate, 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 which causes a proliferation of a particular type of cell, and you wind up dying of this leukemia. And the question is, how can you do something that will lock the kinase domain in a state where it doesn't send that signal? So Gleevec binds in a certain way, and it really changed the whole prognosis and survival statistics for patients with that disease. But resistance does very often develop. Now there are some second-line drugs that overcome that resistance. But the question is, how can we understand that resistance? What's going on structurally with this mutation? What does it say for drug design? So it seems like a promising area as an adjunct to experiments and to trying things out ultimately in vitro and then in animals and then in humans. Another one is the one I illustrated with SHIP2, which is to look at a protein and try and identify new potential binding sites, ones that don't show up if you have a short MD simulation and can't be captured in crystallization, things you just never see. And then especially novel allosteric binding sites, ones in a different place that could affect that structure. Allostery, I mentioned it a couple times, but it's worth saying why that might be of interest. One of the reasons is that very often you'll have the active site be conserved over a large number of proteins. So for example, in kinases, again, the ATP binding site is something that looks pretty similar in a large number of other kinases. And so if you target one, you often have crosstalk where it's, you're also targeting a bunch of other unintended kinase activities. But it appears that in many cases, if you bind to an allosteric site, those are not conserved as much across other molecules of the same class, other proteins of the same type. And so that may be a way of getting better selectivity, where you attack this if you want to turn off a particular kinase without affecting the others. That's just one example. So, this is getting more speculative, not so much in the sense that it may not be useful, but there may be better ways to do it and more accurate ways to do it. We just don't know. But one is predicting binding affinities, it's worth saying that in addition to these theoretical methods, and I can't remember whether I said those are thermodynamic methods and you don't have to see the process. You have to sample intensively and it's really hard and we don't have perfect accuracy, but you don't need MD for that. And the other thing is that binding affinities often are very easy to measure experimentally, so there's no point in doing the computational activity. So sometimes it may be very useful, sometimes not. And then the last one I thought I'd mention, but except in limited circumstances, I don't think that MD would be useful, and that's predicting toxicity. So the thing you might want to look at is whether a given drug binds to not just another in the same class, but binds to any sort of random protein in the body where it's likely to gum up the works. Now, the problem is that there are tons of proteins in the body and other non-protein anti-targets, so it'd be very hard to look for all of them. There are a few usual suspects. So, for example, many drugs are metabolized by cytochrome P450 isoforms in the liver, and if you actually were able to say, this one is going to make it difficult to metabolize certain other drugs, you might say, that has some disadvantages. There's also, if you interact with the HERG-A-GO-GO protein, you can wind up with a problem where you have a prolonged QT interval and have a potentially fatal syndrome called Toussaint de Plante. So if you could, and that also shows up very frequently. So for some of those, you can imagine screening against a few of those known binders, but lots of toxicity comes from other targets in the body, and often, probably most of the time, it's unknown. It's unknown what the target was and how it did it. It just kills patients or causes horrible side effects. But who knows, maybe there's some role for MD there. Just want to end this part by saying this is not at all a substitute for experiments. I gave a few examples where experiments are probably better than simulation, but even where they're not, when we work both scientifically and also now that we're beginning to collaborate with experimentalists who are doing drug discovery, what we usually find is it's not as clean as we come up with something and we make a prediction and somebody else tests it to see whether it's right. Often what it is we'll come up with some idea, we'll talk to somebody who's an experimentalist who knows how to assay various other things, and often who knows more about the target than we do, because we're very broad. We look at a bunch of different targets. The people who use the Anton and the National Resource for Biological Supercomputing, those 133 research groups, some of them know a lot about particular targets, but when we collaborate with experimentalists, most of the time we know less about that particular target than our collaborator does. So they'll often say, hey, could you look at this? We know this happens, but we don't know why, and we need to know that before we can take it to the next step. Also, and this is a very frequent, although somewhat humiliating experience, where we'll come up with an idea, talk to an experimental collaborator, they'll look at it and they'll say, oh, yeah, I see what you're saying. I think you're probably right. Nobody cares about that. That's just not interesting. It doesn't explain anything. But wait, we had an experience with John Currian where we gave him a movie, he looked at it, and he said, yeah, that's not it. Wait a sec. Could you just rewind that part? And we went back and we ran it again. And to me, it just looks like little balls moving. I actually have trouble seeing a lot of these biologically interesting things there, more than many people in our group. But John said, I think, how far is that? I think that may be folding a salt bridge. If it is, that's relevant because of something else. So then we can go back and do experiments. We'll suggest something. He'll say either, yeah, that's known, or, people don't know that. Let's see if it's right, and things like that. So that's the kind of interactive work that we've tended to find most productive. And my guess is it'll be especially true in drug discovery, where that's a finely honed, well-oiled? I don't know, I always mix these things, but where it's a known process that people do all the time in order to make money. So those kinds of collaborators are good for us. So let's see, limitations. I'll just end with this, it won't take too long, but it's very significant. what's wrong with MD that we know about, and then there are other things we know don't give the right answer, or we don't know the source. So we do know that we can do a lot of things that are interesting, but even with the various paradigms I outlined for you, most of them don't work all of the time. So there are a couple areas of limitation, and it's hard to tell them apart sometimes. One is, even though Anton runs extremely long simulations compared to previous computational technologies, inadequate sampling, just not being able to run long enough, is clearly a problem in some cases. We can say, okay, we know that's the problem here. And in addition, and more significantly, at least in terms of, for me, the things I find scarier are problems that can be attributed to inaccuracies in the force field. That is, inadequacies in the underlying physical models for the phenomena that MD simulations are designed to explore. So I'm going to give you some examples of that. This one is, let's see, this is, a PDZ domain. I wrote down what it is because I'd actually never heard of this one. It's neuronal nitric oxide synthase, a part of that protein. But the significant thing is that this is known to be a stable protein experimentally, but if you simulate, what you're going to see is it's long enough to see these things, but this helix falls apart fairly quickly, and the functional behavior of this just turns out to be completely wrong. So it's something where we know there's at least a local free energy minimum, and probably it's a robust native structure, and in simulation, repeatedly, it gives the wrong structure. It's not stable in simulation. Now here, this one is, this is the classic model for a disordered protein that's been studied a lot. And what it is the zinc binding domain of HIV integrase. So it turns out that this domain, in the presence of zinc, is quite stable. It has a well-defined native structure. But if zinc isn't there, it becomes disordered, and in a very regular, it's stable in I don't know if you can say stable for disordered, but it's studyable. You have an equilibrium set of measurables, and what happens in simulation is just all wrong. So just running this here. So what's happening is it's collapsing, and in fact it's collapsing from, so let's see, by a factor of 2, I guess, in terms of reasonable measurements of how compact it is, which is about the folded state. And about the same degree of compactness as the folded state, and it shouldn't. Let me just show you, let's see. Okay, yeah. So on the right, first of all, where does the mistake come from? So we ran some quantum mechanical studies and then tried to narrow things down with various experiments to find out why this disordered state is poorly modeled. And this is just one example. What we found in studies of protein folding was that we were mostly interested in the folded state and the pathway to the folded state. And some of the quantities that we measured were extremely accurate, surprisingly accurate. The order of events we think are quite believable, but in the unfolded state, it looked nothing like the real unfolded protein. And then we found other examples, and we're able to tease out some problems with the force field that seem to be at least a potential culprit, and then correct them, but that doesn't mean we've really corrected the underlying problem, but we learned something from it. So I'm not going to try and back up here, but you may remember what that force field looked like. And I said the electrostatic non-bonded term was the one that took the most time, but at the very bottom, the van der Waals interaction is the Leonard-Jones model for that interaction is a term that's proportional to 1 over r to the 6th minus a term that's proportional to 1 over r to the 12th. That 1 over r to the 6th is not, what it really is the first term of an infinite sequence of even even numbered terms, even exponent terms. So it really should be 1 over r to the 6th, some constant times 1 over r to the 6th plus 1 over r to the 8th plus 1 over r to the 10th and so forth. What we found is that if you look at the conventional water models that are used, several of them, the effect of those higher order terms, which I'd always assumed were negligible, that's falling off pretty quickly, turn out not to be negligible, especially in water, although it shows up in other places. And if you added in some of those higher order terms, especially going to 1 over r to the 8th and 1 over r to the 12th, the behavior normalizes, and you actually see the correct radius of gyration, which is this figure of merit for how compact this is. Now, that wasn't much, we really wanted to have a practical force field that we could use on Anton, and something that has these extra terms doesn't fit on, the hardware isn't designed to do that, at least reasonably efficiently. So what we did is sort of a hack, which is to increase the coefficient of that 1 over r to the 6th term to make up for those missing terms, and then made an electrostatic correction and the 1 over r to the 12th as a small correction also, then ran that, and the behavior of the disordered proteins was much more what you would have expected without much, there was a tiny degradation, but very little degradation in the behavior on folded proteins. Now, does that mean we've discovered what's really going wrong and now we have an accurate force field? No, it doesn't. I'm not sure it's an ad hoc correction because there's something going on, but you could imagine that being the sort of thing where you correct one problem and then another, and it never, with the kind of functional form we use now in force fields, in all the force fields that are commonly used, it really doesn't get to the point where you're converging on something that's basically accurate. So at least we understand the problem, we can narrow down some parts of it, and the result is something, did I even run this? I can't remember. Something that looks better. So this is, the radius of gyration stays at, I think it's about 24, which is experimentally correct, compared to about, I believe it's 12 over here, and then the native state, the folded state, is about 11 or so. I may have the 11 or 12 mixed up, reverse. Now, as far as which force fields, I'm usually asked this, which force fields the problems were present in, the basic answer is all of them, all of the most commonly used ones. For the simulation that I just showed you, the water model, you have the same bad behavior with TIP3P and also with various 4-point water models. As far as I guess it's not marked here, but the basic protein force field, this is Amber 99 SB star with the ILDN correction. But again, it's not like the other force fields that we've tested or any better. It's not an Amber specific problem. It's more fundamental than that. And then there's something called TIP4P PD that we described that has that correction in it. So this is an example not of, now we've got the answer, but of force fields used in MD simulation. They're not perfect, and it can actually matter sometimes. I'm going to try and tie this up pretty quickly, but this one is it's a crystallographically known complex structure between the phosphatase PTP1B and a particular 8 micromolar binder, which is, it's not like a super drug, but 8 micromolar is very stable. That's a high enough affinity that, this is a good binder. And yet in our simulation, this guy is going to fly off pretty soon, which it shouldn't. It's very stable. I think it's a little hard to see because it's too fast in the first state, but what it's actually doing is it's changing shape in the bound, so it's already changed shape, and then that allows it to eventually dissociate from PTP1B. Now, the question is, what's going on here? From the studies we've done so far, it looks like the problem with the force field is not so much in the protein, it's in the model for the ligand. Now, the ligand was modeled using, it's another amber one, but it's not specific to amber. Once again, it's the GAF model of the ligand, the generalized amber force field, I guess. The problem is that with proteins, for decades now, people have fit all of the parameters within that basic functional form. They fit them to proteins. There are only 20 types of natural amino acids, and there are a few, you've got pi and phi angles, and then you've got some side chain angles. You play around with the various constants. For a ligand, it can be an arbitrary small organic molecule, so the process of finding a force field, a set of parameters that's applicable not just to one ligand or small set of very similar ligands, but to anything you can throw at it, is very difficult. What you find is difficulties in what's referred to as transferability, where you try it on something else, it just doesn't work. Very hard task, and it's surprising how you can get reasonable answers from some of those force fields, but in this case, it fails, and it's known that it's failing that way. Like a simple test was, if you constrain the conformation, sort of a soft constraint on the conformation of the ligand, and start it with that crystal structure, it stays there. It stays where it's supposed to, even though the protein force field is the same. So again, we've got problems. Now, what's wrong with the force fields? Quick answer is we don't know. We've discovered some things, other people have discovered some things, but here's some of the obvious ones. Polarizability is one. Current force fields, the ones that are in use almost all the time, are what's called fixed charge force fields. And those are ones where for every atom, or specifically every atom type, you can say, okay, carbons are different if they're aromatic or aliphatic, and if one of these atoms is connected to these near neighbors, well, that's a different kind. But for each of those atoms, you have a handful of different things where you can say, just based on the structure, based on its covalent neighbors, here's what atom type it is. But for that given atom type, you're assuming a fixed partial charge on that atom. Now, that's just wrong. In reality, there are simple examples. If you have a given atom here in a structure and some other part of the protein wanders over here that's negatively charged, the electron cloud is going to, get moved to one side or another, depending what those neighbors are. Charge can flow in various places, and the charge is just wrong, and it's oriented wrong. So one thought is, well, what if a thought that's been there for, a couple dozen years probably, is what if we modeled that correctly and we had charges that could be polarized, so it would be off-center from the atom, either a simple thing where you represent a dipole with a finite approximation, or maybe even higher order things, quadrupoles or something. It could give you a sense for what the shape of that electron cloud is. Could that help? We don't know. We've experimented with polarizable force fields with another, I'll just mention the other and say where we are and other people are. Another one is that most force fields represent those charges as fixed point charges centered right on the atom. But that's not right. Like quantum mechanics, well, real quantum mechanically, it's a fuzzy, three-dimensional space, and you can't describe it this way at all. But one thing we do know is that those charges would be better modeled as sort of a smear. If you look at the hydrogen atom, there's an exponential smear of the electron cloud. It's not just at one point. So what if you used diffuse charges and then also added polarizability? We've been playing with the force field, first of all with fixed charge force fields, but also with a polarizable one with diffuse charges. And we've learned a lot. There's some things that are a little bit better, but to a first approximation, it's not a huge improvement in our hands. Other people have worked on it. Again, there are some advances, but it's more expensive. These kinds of computations, at least at this point, can take as much as an order of magnitude more time, at least a small factor. And our group basically said our success would be measured if the chemists and biologists in our groups abandoned current force fields and started using that, even though there's a time penalty, and we're nowhere near that. So we at least don't know we haven't figured out how to fix force fields with just that. Hydrogen bonds are another. Hydrogen bonds, even though there have been experiments with ones that are more proper, typically hydrogen bonds are measured simply in what amounts to electrostatic way, and real hydrogen bonds are oriented. They're preferred angles, between the 4 different atoms that are involved in a hydrogen bond. Is that the problem? We don't know. I mentioned the dispersion term. Is that truncated series an issue? None of this shows up in the Schrodinger wave equation either. The whole breakdown of those force fields is wrong, and there's no obvious way to solve it. You just can't solve the Schrodinger equation. You can't do it in closed form at all beyond the hydrogen atom. And good approximations, it's very, very hard to do, way beyond the efficiency you'd need for long MD simulations. There's some specific ones. Combining rules is another. Won't spend much time on it, but some of those interactions, for example, the dispersion term, 1 over r to the 6th, you take 2, what you have is 2 parameters there, 1 for each of the 2 atoms, and then you do a certain arithmetic average or geometric average. That's not right. Really, you should think of it as at least a 2-dimensional table, which is less efficient and so forth. Anton will support some of those 2D tabular forms, but those are still real approximations. And then I was getting to the basic functional form. What if the whole thing, that whole way we break things down, is wrong? And, there's some things where, electrostatic interactions, 1 over r interactions, do show up in the Schrodinger form, but they're not in a straightforward way. You can't just, it's not really an additive term. It's got all this quantum weirdness. So if that happens, suppose we even figure it out, what if it would be too expensive to solve it? What if there's no good way to implement that anywhere near as efficiently as you'd need to get biologically valid results in all situations? Again, we can see things where it works, but maybe some of those problems we're running into are in practice unsolvable. And even before that, it could turn out to be there's some things where there are better algorithms to solve those problems, but Anton is not going to be a machine that does it. It could be, like Anton can do anything in principle, but if it's 2 orders of magnitude slower, it may be that you're better off using a GPU in the cluster of chips in a more ordinary way. So we don't know if that'll happen. And the final thing I wanted to say there is, just in terms of a consolation prize, what if we can't do much better, we as a field can't do much better in accuracy than we are now? One thing that might be nice is if we can at least characterize what some of those problems are, that might be useful in terms of insights into biology, into biophysics in particular. What theory is a whole set of abstractions where you try and figure out, yeah, it's wrong, Newtonian mechanics is wrong, non-relativistic, physics is wrong, but you can do a lot of prediction. What if we look at this and we say, this model doesn't work, and here are some, maybe we learn, here are some effects that we're modeling that are much more important biologically than we'd ever thought. And my hunch is also there'll be some things where we're going to, find out, we've been spending all this time and effort trying to make this particular part of the force field very accurate, and actually it just doesn't matter. You can do violence to that, do it any number of different ways, and I don't know, you get error cancellation, and it just doesn't matter how you do it. That could simplify things also. And if you're building a theory, if all you're trying to do is understanding biology, what are the physics of biology, maybe that'll turn out to be very interesting, even if we can't get the simulations all right. I believe that's it, so we probably have, I don't know, minus 15 or 20 minutes of time for questions, but if anybody has the patience, I'm glad to answer some of them. Do you want to take this? Okay. Now, I think this is the main one, but I'll give you this one back. I have a tendency to wander off with them in my conversations.
Are there any questions to David? I think she was a little, fraction of a second.
Give it to you next. Thank you. I was curious, when you talk about all of these different corrections that you can do, whether it's changing the van der Waals or the polarization, do you have an instinct for which problems you would want to change van der Waals versus which problems you would want to change polarization or something like that?
Yeah, in terms of which biological systems and so forth, I don't, first of all, or at least at this stage, I really don't. But, you know, the goal would be to have one force field that would work for pretty much everything. Maybe some exceptions, like if you're dealing with nucleic acids, which is a much less mature area. That's an area where not as much work has been done on them over the years, and especially now RNA is such an interesting area, it would be good to have it work there. Well, if you had a force field that worked especially well for those, you would know you're studying those and it would be okay to use it. But in general, you want transferability across all of those different systems. Now, there are some things where, I'm not sure I'd call it insight in my case, but there are things that we know are hard, even though we're not sure how to solve them. So some examples, well, the simplest one would be we don't do reactive chemistry at all. Bonds aren't broken or formed in this kind of simulation. But also some things that have a quantum mechanical component to them, like metals. Metals aren't handled well if you have metal coordination. Protonation state also is something where you can imagine a current force field, you know, saying something about the environment and saying this should be protonated or deprotonated, but we at least just do that by hand. Well, in fact, sometimes it's very embarrassing. This early paper, the sodium protein antiporter that we had many years ago in Science, what we did there is there were 3 different aspartate residues, I guess. We had no idea what the right protonation states are, so we tried all 8 combinations and just ran them, and you know, one turned out to work, where one turned out to be the binding site, one was the regulatory domain, and one did something else I can't remember. And if there are a bunch of those, or disulfide bonds is another one, you know, we'll just do that by hand. We know, you know, there's 2 cysteines, but we don't know what's connected. So those kinds of things we know to be problematic, but it would be very useful to have a sense for what kind of problems we can not trust the results for. If we at least knew that, we could say, this probably is right, and this other one we're not sure, or it's probably wrong, and I at least don't know that. I don't have a feeling for it.
Okay, thank you. Could it be the case that by running long MD simulations, we are just taking a look at the kinetic part of the processes, disregarding the thermodynamic processes?
Yeah, it's an excellent question. So if you ran long enough, if you run for a long enough period of time, then we know that, you know, the whole ergodic principle would say you're looking at the thermodynamics perfectly, but it's not infinite, it's far from it. And in fact, what you do, which sometimes is very interesting, is you'll detect a kinetic barrier, and then eventually it will surpass that barrier. But telling the 2 apart can be extremely difficult. We know we can do a lot more of that than we can with short simulations, but it's not always clear whether we've explored, whether we've sampled the space according to the Boltzmann distribution fully. Now, for some of the I won't say non-standard, but if you were going to move from straight MD simulations to using other sampling techniques, some of them are much better at crossing kinetic barriers. These aren't our techniques. And we do, in fact, use Anton for some of those enhanced sampling methods, which sometimes will have the quality of, you know, one is artificially lowering a barrier so you can get over it more easily, get the correct thermodynamics, but not the correct kinetics. And so you need often an adjustment there of various sorts, but sometimes that can be made rigorous. Or another thing is various techniques that satisfy detailed balance. They're thermodynamically correct, and then involve things like, simplest cases, heating things up, where you take a system and then gradually you run copies of it at various temperatures, and at a certain point, the low temperature one moves up to a high temperature, and at the same time you move a high temperature one to the bottom. You clearly know those sorts of techniques from your nodding. And you can come up with something where if you get back to the ground state temperature, which might be biological or experimental, you'll get a correct distribution at that level, and hopefully in some cases you get it more efficiently. But straight MD simulation is absolutely the case that, first of all, you won't always get the correct thermodynamics, and secondly, you won't always know whether you've got the correct thermodynamics. Very important limitation.
David, have you tried to use your long trajectories to calculate a covariance and then use that to generate normal modes? And compare with normal modes calculated with coarse grain methods, or?
We actually haven't. It's a good question. So, I mean, some of these normal mode analyses are based just on an elastic network model where you don't even need to know. I mean, you get interesting results without even needing to know, electrostatic interactions or any of those sorts of things. Just with, I guess it's certainly with bond length constraints, and I can't even remember how they often deal with bond angles or torsion angles.
Well, you can use the same. You can use the same potential actually.
Sorry, the same potential?
Yeah, you can use the same MD potential.
Yeah, right. So that's a very interesting thing to study if you've got a lot of data. And some of those are very beautiful. You look at the pictures afterward, and sure enough, one of these normal modes picks up this hinge action, and this other one picks up this biologically interesting sorts of effect also. But we haven't done that.
Any more questions? I think there was one at the back earlier. I'll come back to you.
So we always run these simulations in water, but we know that in a cell it's going to be a very crowded environment and you have all this other things happening. Do you think that that would affect the folding and dynamics that you want to study? And do you think that something like Anton might eventually get to the point where we can actually test that?
Yeah, that's a great question. It's a fascinating thing. My sister, Suzanne Pfeffer, who's chairman of the biochemistry department at Stanford, I remember years ago when I became interested in, folding, and, I described the whole thing just as a computer scientist would, and she said, that's not what a cell is like. It's not water, it's jello. And in fact, it's not really even jello. It's a bunch of these things all bumping into each other. I had no idea. And now there are people doing excellent studies, experimental studies anyway, of the effects of crowding. Now, some of those things happen slowly. They happen, at a level where MD simulations would have to be very long. And I know there's some other things where people have done some computational work that bore directly on the question and that went beyond, simple viscosity constants, manipulating those. But I don't know much about them, and we haven't done that. It's a really great question, though. It may be that some mechanisms are dependent on a crowded environment.
Yeah, thanks for a wonderful talk. So to me, the biggest thing I actually take away from your work is that all of these issues that you described notwithstanding, when you actually take a force field, an off-the-shelf modern force field, and you turn on, you basically press go, that a good number of the time the protein actually folds. And sometimes it doesn't, and there are issues and so on. And sometimes the thermodynamics isn't right, sometimes the kinetics isn't right. But there's no reason that should happen, really, because
That's
it's exactly not like
right.
these force fields were ever designed to fold proteins. They were built bottom up. So that, to me, means something very deep about some sort of robustness in the system. So I'm just wondering if you could say something about that.
I say that almost word for word in some of my talks. That's right. When I look at this, sometimes I'll say, why on earth does this work at all? It's so wrong. And also, if you look at the size of some of the individual errors, they're just way out of proportion to, there are many KT. They're just all wrong completely. Why does this happen? And then the next part I also think about a lot with what you said, which is there something deep there? something that says that robustness is built in by evolution. And I don't know enough to know the answer, but I know sometimes I feel like, yeah, that must be right. And, it shouldn't be as hard as we think. We shouldn't be so depressed because nature wouldn't set it up in a way where having a little crowding or a little push or something like that would make the whole thing not work. And then on my bad days, sometimes I'll think, well, wait a sec, maybe actually nature wants it to be exquisitely balanced and we need to have that. So an argument for that might be, we do know that some things are energetically very, very balanced. So you'll have a hinge motion, for example, where the barrier would be too high if it had something that stabilized things too much over here. So maybe it wants it very carefully balanced, and if everything doesn't come out, exactly right, that can flip it. And if nature does it for a long time, it may not care, it just wants the statistics right. So that would be bad news, because then you could imagine that even with all the things that do work, we could work on this forever, and we're not going to get it right enough. So I don't know, but it is kind of amazing that it works at all. Yeah.
All right, on this positive note, we will close the session. We will thank David for a wonderful talk.
Thank you so much.
And I know there are many more questions, so if you can grab Dave's attention, run quickly to the podium and be
Or
the
somewhere.
first to
Let me
ask
put
him.
this down. Thank
So
you.
thank you very much.